research in decision making under uncertainty in medical research.
Thanks, Shipra, and thanks for the organizers. It's an opportunity for me to tell you about some of the things that are happening in decision making and clinical research. The outline is I'm going to tell you about randomized trials. You probably don't know much about them. Most people don't, even patients in them don't. I'll tell you about sequential decision analysis and multi-arm bandit problems. I know that many of you know more than I do about them. And there's some sessions in the workshop dealing with them, but I'll give you my perspective. It'll be pretty elementary in developing them and taking you back a few years. My efforts to interject and inject decision analysis into clinical research. It's been a frustrating experience for the past 40 years trying to get people to do what I think are reasonable things in clinical trials. There have been some successes. Naturally, I don't know naturally, but I'll focus on the successes. But this is a work in progress. It's been at least 40 years going on, and there's some progress. I'll show you the progress. But we continue to try to do better things, and biology is making us do better things. And that's where you come in. And that's the reason I'm here, is to tell you about this and to get you to bring your brains and your brawn into the problem. Along those lines, I'm helping to organize a meeting at the New York Academy of Sciences with this title: The Need to Accelerate Therapeutic Development, Must Randomized Controlled Trials Give Way? So we had a meeting, we were scheduled to have a meeting this morning, and this is a note that I sent to the other members of the organizing committee. I said, I can't make the call, but by coincidence, I'll be giving a presentation at the workshop, this workshop. My goal is to show computer scientists how optimization and decision-making under uncertainty applies to clinical trials, and how, if in fact RCTs do give way, your expertise will help identify better and maybe even optimal replacements. So what are randomized clinical trials? In the 1940s, the state of clinical research was anecdote case studies, published case studies. For the previous almost 200 years, some people had been trying to get, evidence-based medicine that we still hear about today. But it was in the 1940s that there was this transformative idea to take an idea that was originally R.A. Fisher's in agriculture and bring it into medicine. It was Bradford Hill that brought it into medicine. It was randomization. A pretty simple notion where you eliminate bias, assignment bias of patients. Sometimes a patient in the past was given a particular therapy because the doctor thought that was a pretty good thing for that patient. We didn't learn much. In the 1940s, originally in a trial of tuberculosis and streptomycin, Bradford Hill randomized patients. So if you came in, they flipped a coin, and half the time you got streptomycin, and half the time you got nothing. And then they saw what the results were. And the results were positive. But that led to that innovation being used through today in almost the same sort of circumstance and almost the same sort of sophistication. 50-50 randomization. Sometimes everybody's blinded, that's dealing with the patient. There's a statistician who knows what the assignment is. But, and then at the end of the trial, they open up the envelope and publish the results. And increasingly, trials have gotten bigger. We've run randomized trials, 10,000, 20,000 patients. And in cancer, if you know anything about what's happening in the world of cancer biology, where with the genome, with the various markers, we identify that this cancer is driven by such and such oncogene, one of many, and one of many in the same tumor, and that's the cleverness of cancer and the reason we haven't cured it. And the biologist builds an antidote, builds an antibody against that marker. What it means is that we used to have breast cancer. Now we have HER2 positive, estrogen receptor negative, AKT positive, PI3 kinase positive, finer and finer subsets of the disease, such that we no longer have 100,000 patients with, or 200,000 in this country per year with breast cancer. We have these smaller and smaller diseases, and soon we'll be treating, every patient will have what's called an orphan disease. And we don't know how to develop therapies. In the context of the randomized trial, the experimental unit is the trial, where at the end of the day, you calculate a p-value and you say this was statistically significant or not. Now, when I was a graduate student, started working on what I thought was a clinical trial, bandit problem, I figured that this was the way clinical research was conducted. When I got into, after I published my dissertation in 1972 on a Bernoulli two-armed bandit problem, I thought this was a great thing for clinical research. I thought this was the way clinical trials would be conducted. And I was aghast to find that not only did they not do something like this, they never looked at the data. So in a clinical trial that's running, you design the trial, 50-50, open up the envelope, and see whether it's positive or negative. You're addressing one question. And increasingly in the world today, we can't address one question and make headway. And indeed, that one question, not only is this a 5,000 or 10,000 patient trial, but it takes 7 to 10 years to run. We can't do that. 7 to 10 years from now, the therapy that we think today is great is going to be outmoded. And so progress is incredibly slow. And we need better procedures, and procedures where the experimental unit is the patient rather than the trial. So in 1985, I published what was at the time a pretty comprehensive assessment of what bandit problems were, including not only in statistics, but in engineering, in computer science, and psychology. And there's an annotated bibliography that discusses each of the papers that we knew about at the time. Things have moved on. Just to show that I've done some things in bandits. In 2002, and the date is not important, somebody did a survey of physicians and asked, so why do we do clinical trials? What are we trying to accomplish? And they reported that many respondents viewed the main purpose of benefiting participants rather than creating generalizable knowledge. This view, which was more prevalent among specialists such as pediatric oncologists that enrolled greater proportions of patients in trials, conflicts with the established principles of research ethics. The established principles of research ethics are contained in something called the Belmont Report. And it says very specifically that we can't, when in a clinical trial, if you're in a clinical trial, you're a guinea pig. We're learning. We're not trying to treat you. The goal is not to treat the patients in the trial. And this to me, and maybe it was appropriate back then with the randomized trial being the standard, the trial is the unit, but it's not appropriate today. And you see that there's some pushback among oncologists as to, is this the appropriate thing to do? So I've been trying on various fronts to change this over the course of time, and more recently, going into the lay literature. And this is an article in Fortune magazine where I talk about what traditional trials are and how they don't accommodate the amazing changes in biology. The trials have sample sizes in the thousands, even though increasingly the number of patients with the disease are in the hundreds or less. Even if we could do a large trial, its results will be irrelevant when they're finally announced. The patient should be the unit of inference. We shouldn't be doing p-values, we should be doing patient values. And we need to merge clinical research and clinical practice. So I spent a lot of time trying to persuade my colleagues in biostatistics and in medicine to do other things, to look at the data and adapt to what's happening in the trial, and to modify the course of the trial on the basis of what you're seeing. Not only stopping early, but adding drugs, dropping drugs, looking at who's benefiting from what, trying to get greater precision associated with that subset, et cetera, things like that. And it was a stone wall. And so I decided, and I was continually told, Barry, you're naive, you don't know what the facts are, this is not the way we do things, and get out of here. So I decided I'd become a mole. I'd actually try to learn some of this stuff. This was a profile science did on me back a number of years ago, and one of the statements is, Barry would be the lead statistician for the CLGB is a cooperative cancer group. It's the cancer and leukemia group B. Breast cancer studies. He was not greeted warmly. I objected rather strenuously, recalls Craig Henderson, a breast oncologist at UCSF, who had heard that Bayesians were loosey-goosey in adhering to the rules. Suffice to say that I converted Craig Henderson, and he became somebody who, even through today, is advocating doing adaptive things. And this is just to show you that I was successful on that side. This says ranking in specific field, highly cited researcher in a specific field, and the point is that this specific field, that's an emphasis under the thing, is clinical medicine. So I'm known in clinical medicine. What that means is, it used to be, when I started out this CLGB thing, I'd suggest some radical things, and people would say, you know, well, that's not what we usually do. Eventually, and with my increasing acceptance, and this is a message for you if you want to have an impact on a particular substantive area to become a mole, eventually they said, well, Don says it's okay, so we can do this. And I'll show you some of the amazing, ludicrous things that we did. So a two-arm bandit. What is a two-arm bandit? Eventually we do, of course, many-arm bandits. You pull an arm, you win or lose. My focus was Bayesian, as you gather. You have two arms, two coins. Probabilities of heads on the two coins are not known. They have a probability distribution. In the Bayesian approach, anything you don't know has a probability distribution. So let's assume we're starting out non-informative prior on both of the Bernoulli parameters. Back here, I called them left arm, right arm. I thought that was pretty cute. So rho is the right arm, and lambda is the left arm. But here I'm changing it to A and B. So you have a probability distribution on A, a probability distribution on B, and you get to pick one of them to start with. What do you pick? Well, of course, it doesn't matter. If you tell me A is optimal, I'll say, well, B is exchangeable with A, and therefore it's optimal. So they're both optimal. So you pick one. You pick A, and you win $1. The probability distribution changes, and if you get a success, then you are in this mode. If you get a failure, you're in this mode, and of course you're comparing to the same, arms are independent, so you're comparing to the same distribution from before. What to do? Well, if you read that thing about my dissertation, or even if you hadn't, you certainly would say, let's do, if we got this, let's do it again. Let's do A again. If we got this, let's switch and do B. So you choose A again, say, after you get a success, and you get a loss, and now you're in this situation. So now what? It's not so easy. They both have the same mean, but they have different variances or different variabilities. So there are different reasons that one might want to do this. Well, what are you trying to do? And so let's set a context for what you're trying to do. Suppose you're going to run a clinical trial of length n, little n, and at the end of the trial, you're going to use the one that won. And you can do adaptive things during the course of the trial. And the objective is to maximize the expected number of successes over the total patient horizon, which is a number bigger than little n. It's the number of patients who have the disease. We don't know exactly what it will be in the future, but it doesn't matter much. And as you see, what really matters is kind of an order of magnitude of capital N. And this is something that was put forward amazingly in the same issue of the Journal of the American Statistical Association, 1963, independently by two great statisticians, Ted Colton and Frank Anscombe. And they talked about, how to build a clinical trial. They didn't win the battle because they never had any influence on what was happening in clinical research. And the goal is to treat patients effectively, back to the Belmont report, whether they're in the trial or not. We don't want to sacrifice the patients in the trial. We do want to learn from them, and recognizing, people have said to me, well, yeah, but you're learning from the patients who are in the trial, and the patients at the end of the trial may get a different treatment or a different probability of getting different treatments because of what you've learned, and you're treating the patients badly in the early part of the trial. And my response is, it's always good to delay getting sick. So you're subject to, the uncertainties in the early part, and if you could wait a couple of years to get a disease, you'd take benefit of anything that happens in the meantime. So the solution is by dynamic programming, as you all know. This is today. If you use arm A, you move to do different things tomorrow, either state after you fail or succeed, like we saw in that earlier example. If you use arm B, you go down, and the same kind of circumstance. Now embed that into a bigger picture. The clinical trial in the example is length 7. And so you go out to set these. This is a complete description of the possible states starting from today and going out till you get 7 total observations. And this is along the way, the number on arm A and the number on arm B, and then the various possibilities within these boxes. So here is everything that can happen at the end of, if you're dumb, you're not doing anything clever, you're just seeing the possible states after 7 total observations. And one of these, for example, so this is where you've got 5 observations on A, 2 on B, and the number of successes on A, the number of successes on B. So 18 possibilities at the end of the day. And the numbers I put in here, I'm assuming capital N is 100. So at the end of the 7, there's another 93 patients that you treat. And you're going to use the one, according to the rule I set up, the one which had the better response at the end of the 7 for those remaining 93. 7 is not a magic number here. It's just, something I can show on the page. And these numbers I've written down here are the numbers of successes in the subsequent part of the problem in the additional 93 patients. So, for example, if you've observed 5 out of 5 successes on arm A, it doesn't matter what you observed on arm B, because you're going to use arm A for the remainder. And your probability of success on the remainder is just Laplace's rule. It's just 6 out of 7. So take 6/7 of 93 and you get 79.7 or something like that, rounded off to 80. And so this is, this is the end of the trial, the experimental part, and we write down it for every one of these possibilities at the end. And then we back up. For example, this is after 6. This is one of the many possibilities that you could have. It happens to be 4 on A, 2 on B, and you had 3 out of 4 successes on A and 1 out of 2 successes on B. If you go to the right, you go to one of 2 different distributions, 2 beta distributions. That's what happens if you get a success. You start with beta 4-2, and you move, this is on arm A only, and you move to beta 5-2 or beta 4-3, depending on whether you get a success or failure. And the neat thing about the Bayesian approach is you can predict. You can say, what is the probability of these individual things? And that's, again, just Laplace's rule. So there's a 2/3 chance of success, a 1/3 chance of failure, and so you can calculate the probabilities. For example, here, you're going to go to one of these 2 places, and this one with probability 2/3, this one with probability 1/3. Take the average of those 2 things and plug in that number here. And then fill it out. Keep going back to the beginning, and what I've laid out here is not the numbers, but which arm you used. Which arm was the one that gave the maximum for each of the states. And so what you see here along this line, as you knew from the beginning, since they were symmetric, it didn't matter what you did. So they're both optimal. It doesn't matter which way you go. But when they're shaded in, you should move down. And when there's no shading, you move to the right. So back to this case. That's a point here. That's that point, and it says you should move down. It says, as many of you knew, this would be the appropriate thing to do. Why is that? Intuitively, there are two desiderata. One is you want to make money, or you want to treat the patient in front of you effectively. And the other is you want to get information that's going to help you into the future. And those two things make the problem what it is. I mean, make the problem incredibly difficult to, prove things about, for example. And so this is what you do. And if you do write down the numbers, you'll find that the number that's written in at the beginning is 63. 63 is the expected number of successes of the 100 patients using the optimal strategy that has this constraint that you can be adaptive only in the first 7. Now, if you compare other kinds of designs, for example, this is the dumb design, the n equals 0, you don't do any experimentation. You either use arm A forever, arm B forever, any kind of in between, and you're not looking at the data. Then you get just the mean of the two distributions. So 50 is the dumb answer. It's also the answer for a randomized trial, by the way, if you do 50-50. And our answer was 63. If you did the optimal adaptive design from the beginning and did, let the small n be equal to big N and just use the optimal one at every step of the way, you get about 65. The maximum that you can get is 66 2/3. That's just the so-called expected value of perfect information. If you have a uniform distribution on the square, and if I'm going to tell you what the answer is, one second. If I'm going to tell you what the answer is, that is which one is better, what is your expected value of what I tell you? It's the expected max of those two things, which is 2/3. Yes, sir.
Does this require you to know immediately whether or not your arm was a failure or a win, which you have in multi-arm bandit settings, but not necessarily in the medical trials, I imagine?
So no, this is you're starting at the beginning, and you've got this random variable, random distribution on PA and PB, and I'm going to tell you which one is bigger. How much is that worth to you? Well, you're going to be able to proceed with certainty that you've got the right answer. What is the expected value of the information that I'm giving you? It's this 2/3.
But what I'm asking is, in the actual medical trial, like you have 100 patients, and patient 35 gets arm A, and now you have to, don't you have to pull arm 36 before you know the answer for arm 35? That's my question.
Yeah. So that's a very realistic and important problem. And you're right, I should have said this immediately, that I'm assuming you get immediate responses in the adaptive part here. And actually in some examples, in migraine, for example, in migraine, the so-called registration endpoint, you can get a drug approved for migraine. on the basis of 2 hours pain-free. So you give the drug, and either the migraine goes away or it doesn't. So you're getting the information right away. If you're treating horrible disease like pancreatic cancer and the endpoint, the outcome of interest, is mortality, even though they die pretty quickly relative to the rest of us, it's still, a matter of months before you see the answer. This raises a very important thing that we're doing, and in your modeling to take into consideration. Take that pancreatic example. The patient's still alive after 6 months, but the patient has characteristics. The tumor has grown, the tumor hasn't grown. The patient is in bed and can't get out. The patient is playing his second 18-hole golf course for the day. There's information that you're getting along the way, and it isn't just waiting till that final endpoint. This is an issue that I keep pushing everybody on, and it's a hard thing to sell because I don't want to go into it. But it's a very important issue from the point of view of adapting, because we want information to adapt to, but it's also important for understanding the disease. And so building these longitudinal models of disease. And it's one of the shames is when we publish papers in cancer, this traditional randomized trial, A versus B, we say what the survival is for A, what the survival is for B, and we compare them. We say what the progression time was for A and for B, and we compare them. We say what the response was for A. This is a tumor response. A tumor decreased in size or didn't. Was for A and for B. We give other characteristics of what's happening, but it's all marginal. You can't put it together. It's only, you need to get the raw data to put it together, but nobody does that because they don't appreciate, the information aspect. Thanks for that great question.
Yeah.
Yeah.
So are you saying that you think of surrogate variables, you think of in the RCM, Rubin's Column model type of thing? You're saying there's no integration of those concepts with the more dynamic model, multi-arm type of thing? Because you have principal stratification and so on, right?
So I'm not sure I understood everything, but you said the word surrogate?
Yeah. When you have a surrogate variable of the type you're mentioning in disease progression, then you could use the Rubin's Column model, and you could have methods like principal stratification to handle all those issues.
Well, I mean, they're all, time varying covariates, there are all kinds of things you could do. I'll tell you what I do in some of the trials I'm going to be showing you, is I condition on the information that's going over time. So not only do I get the final endpoint, but I get the assessment of the MRI in breast cancer, where we leave the tumor in the breast and we get an assessment over time of how big it is. So I use that, and I have a model, a longitudinal model that has parameters that I don't know, that I update, including depending on treatment. And then I calculate the predictive distribution for each patient in the trial. And the usual way that I do that is multiple imputation. So I have this model, and I impute for each patient what the eventual outcome is, and then I do it again and again and again. That gives me, the overall probability distribution. Excellent point. So an RCT for 7 gives you 62.4. I mean, you are getting, 3 on one and 4 on the other one, or vice versa. An interesting thing here, a theorem, if you'd like to try to prove it, is for an RCT, n odd is actually better than getting one more patient. This means to me strict better. So 7 is better than 8. It's better to experiment with 7 to constrain yourself to a smaller number in this randomized trial, which of course doesn't happen in the adaptive setting. So I'm not going to say anything, I'm not going to show anything about this, but if you compare the Bayesian adaptive approach in the scenario I just gave you versus an RCT or versus something like the play the winner rule. The play the winner rule is whatever you got as a success on the last one, you if it was a success on the last one, you use it. If it's a failure, you switch and use the other one. This point of this is simply to say that even though the Bayesian adaptive approach is optimal overall, that is on the average with respect to the prior distribution, it's also better than these two and many others point-wise. So regardless of what the truth is, it finds out the truth. Certainly in the large sample setting, but even in the moderate sample settings. So decision analysis of trials, list the possible results, calculate the predictive probabilities of each of the possible results. This in itself, if you work with clinical trialists, is an innovation. People do things for the strangest reasons, and if you ask them, so suppose you do the trial and you get this. What would you do next? And they say, well, I would do such and such. I say, okay, suppose you get these data instead, what would you do next? Well, I would do such and such. The same such and such? That is, you do the same thing no matter what? Then why are you doing the trial? So write down the possible answers and write down the implications of those answers. That is, evaluate the utility of those results and the predictive probabilities, and then average. That gives you the average utility of the strategy, or in the decision analysis term, the utility of the strategy. The utility is its average with respect to the probability distribution. And compare utilities for the various strategies, for the various designs, and use the one with the highest would give you the optimal, but sometimes for logistical or other reasons you might use something that just was pretty good. Choosing a sample size. Again, the utility is patients in the trial, treat patients effectively in the trial and after the trial. And again, the same sort of setup that I was doing before. Patient horizon is n. It's enough to know its magnitude, I say. So this is an example, decision analysis example. How big should a trial be? And now, even if this is randomized, a decision analysis of equally randomized trials. And to kind of set the context here, I said, suppose you're going to do one trial and then use the one that's effective, the treatment that's effective in that one trial, whether it's a 2-arm trial or a 1-arm trial. And let's suppose that if there were 1 million patients that you're trying to treat effectively, something like coronary artery disease, and the optimal sample size is 1,000, how big would it be with the same setup, the same probability distributions, but with a smaller sample size? If the population size is only 100,000, then it's, a third of what it was here. You'll notice in looking at these that these are just the square root of these. If you had 2 trials, that is, you do one trial, look at the results, do a second trial, and you know that that second trial can have a sample size that you choose based on the result of the first trial, how big should it be under the same scenario which gave rise to this 1,000? And the answer is the cube root, all the way along here. So we actually proved that. And we, of course, conjectured that if you had 3 trials, that it would be the 4th root. But we couldn't prove that, or we couldn't prove in general that result. The point is, and if you show this to a frequentist biostatistician that's drunk the Kool-Aid, this is irrelevant. The way they design trials is they pick a clinically significant difference, control type 1 error, have 80% power, and that's the answer. The population size doesn't matter. So you do the same thing in breast cancer or cardiovascular disease as you would in relapsed Wilms tumor, which is a tiny, tiny, less than 100 patients per year in the United States. You'd do the same thing. What does that mean? You can't do 1,000 patients in relapsed Wilms tumor. So if you go to the NCI, or National Cancer Institute, with one of these cooperative groups, and you say, I want to do this trial in relapsed Wilms, I know because I've done this, in relapsed Wilms tumor, and I want to look at a small number of patients and learn from that number of patients, like 20 patients, and move on. You can't do that. I said, why can't I do that? Because you don't have enough power. And it's this artificial thing that somehow, you can, and so what does that mean? There are no learning trials in relapsed Wilms tumor because they won't fund it. And it's a niche disease, and so pharmaceutical companies aren't interested in doing a clinical trial, especially in view of the attitude. Things are changing, but that's the current attitude. I want to get on to some other stuff, so I'm going to skip this. Just to mention that back in 1933, even older than I am, Thomson suggested a neat strategy, which is not an optimal strategy in any sense, and he says it's not optimal. It's not the best possible, but it seems like it would be pretty good. What you do is, at each time, it's a Bayesian approach, what you do at each time is you calculate the probability that A is better than B. And then you assign the next patient with that probability. So that's an adaptive randomization. It's not optimal, but it's not bad. It's what Google does, by the way, in the search engine stuff. And it's what I've done in a number of settings. And I'm going to, this is one example, and I'm going to skip that. See, I do give you cautionary tales where the result of this adaptive trial that we did, using something like the Thomson approach, nobody believed it. The company didn't believe it, and so they ran another trial, a so-called real trial, and they ended up killing lots of patients because they didn't believe it. And they're coming around. So, and just to say that there are things you can do not only in the context of treating patients effectively, but getting information. This is an example of a diabetes drug where there are 7 doses, so 7 arms, actually more than that because there's control arms. And you want to adaptively address the question, what is the dose response of this drug? And this one was very successful. I-SPY 2 is a trial that I designed in breast cancer. Here's a cartoon explaining the trial, And the big deal here is that the population is heterogeneous, explicitly so. So we look at various biomarker characteristics in the population. And we have, in this particular case at this time, we're adaptively randomizing 5 experimental arms in standard therapy. We're up to 11 experimental arms now. Arms come and go. Trial's still going on. So we might at one time identify this subset as responsive to arm 2. We might drop arm 3 because it's not doing very well. Identify a different subset that responds to arm 5. We calculate type 1 error rates and all of this stuff, by the way, which is not so easy as in the 2-arm setting. And as time goes along, we fill in other arms. And the goal is always looking forward. It's, we want to identify a subset and a drug matching the two that have at least an 85% success rate in a phase 3 trial, predictive distribution. The goal is to have a small phase 3. And just to show that we're making some progress, this was 2 months ago in the New England Journal of Medicine, a paper coming out of this trial. This is one of the therapies that graduated in a particular signature. And not only one, but 2. This is kind of unique for the New England Journal of Medicine to have 2 tandem results for the same trial. And not only that, but an editorial by 2 MDs and also a perspective by 2 statisticians. So 4 articles in that one issue of the New England Journal of Medicine dealing with I-SPY 2. So it's, you know, we're making some headway. And we give Bayesian distributions, and we say for each of the possible signatures, these are subsets of subtypes of the disease, what is the report card? But also, what is your major? Where is the one you graduated in? And for neratinib, the graduation was in HER2 positive, hormone receptor negative breast cancer. The thing I want to say about this is merely that of the 11 drugs so far that we've looked at, fully 5 of them have graduated. Breast cancer is a disease that is highly sensitive to therapies, a number of therapies, hormone therapies, chemotherapies, targeted therapies. And so these 5 have graduated to phase 3 with different sample sizes. Why? Because we're looking constantly and we're saying, are we there yet? Do we know what the answer is? And we're adaptively randomizing. Neratinib graduated, but along the way, the Data Safety Monitoring Board said to me, you know, Don, this is a hard drug to give, and it's not doing very well in this subset of disease. Maybe we should amend the trial and drop that subset. And I said, you don't have to. Look at the randomization probabilities. The algorithm has already predicted that and is giving that patients in that subset neratinib with probability 0. But meanwhile, it's doing very well in this other subset. These are the goals. And I want to tell you finally about GBM Agile. This is a trial. I just came back from Australia. That's one of the countries. China is also in GBM. GBM is glioblastoma multiforme. It's the brain tumor that killed Beau Biden, Ted Kennedy. It's the most common brain tumor. It's also the worst brain tumor. And nothing works for it. So we're building an international trial. And I want to show you something about it. This is the way it works. New patient comes in. I say it's I-SPY 2 on steroids. It's a registration trial. The I-SPY 2 was a so-called phase 2 trial predicting for phase 3. This trial is actually including phase 3 in it, in a confirmatory subset. So a patient comes in, gets randomized according to the adaptive randomization. We update the patient data, we update the longitudinal model, this thing I talked about before. We calculate the probability that the arm is better than control, and we make decisions. One decision is to stop for futility. Another is to stop because we reached the max of 150 patients in the stage 1. Another is to continue in stage 1. And another set of possibilities that you can graduate. If you graduate, then you may move into, depending on what we decided about that therapy in advance, into a stage 2, which is you get a fixed randomization for the next 50 patients. And you could stop accrual and give it back to the company. And then we continue and add new experimental arms if we can afford that in terms of the accrual rate of the trial. We update the probabilities according to subtype, determine the randomization probabilities, and keep going. So it's like I-SPY 2, potentially never ending. It may serve as the way to process these. Simulations are required. And a consequence of inadequate simulations in clinical trials is similar to what this particular example of inadequate simulations at SFO. So the last thing I want to do is this. It's like do I have 4 minutes? I'm down to 0. Can I show a 4-minute video or not? You've seen it before, Shipra, so what do you think? Is it worth the 4 minutes? There's a danger associated with it, and that is, what's the danger? I'll do this.
Bottom left corner, there's a little play button.
What is it?
Bottom left corner, there's a little play button. When you hovered over it, there's a little, yeah.
There,
see,
yes, sir.
No, I just click. Oh, okay. A danger is that you may change your vocation and want to go into
senior scientists,
this really
clinicians, and supporting
stirring
foundations from multiple disciplines and countries met as part of an unprecedented international collaboration, taking scientific crowdsourcing to a new level.
An Australian
what began as a small group and a transformative idea
foundation
rapidly grew to include 130
that
contributors.
is an advocacy group
So
for
what is
brain
motivating
cancer.
them to forgo personal
And so they
interests,
put this together.
to unite and work unpaid?
They just listened to what I was
The
saying
answer
over time
is to develop
and
an
put this together, and they did a great
global
job.
adaptive clinical trial to tackle one of the world's deadliest diseases, the malignant brain cancer glioblastoma multiforme, or GBM.
We're here to do something that's different, something that is historic.
Dr. Timothy Clousey, the principal investigator for GBM Agile, says it is more than a clinical trial. It is a learning system that offers unprecedented opportunities to identify effective therapies for GBM.
I'm always amazed when we get a group like this together, of this many real leaders in this field, to begin to listen to each other and change their mindset, open up their minds to new ideas. And that's what this is about, actually.
I think what we need to keep an eye on here is not curing the tumor or running the trial or any of that. What we need to keep our eye on is that this affects people.
That's what adaptive trials are bringing. Faster drug development, more efficient, more accurate, and hopefully bringing better therapies to people.
Traditional clinical trials typically only test one therapy against the standard of care. They follow a one-size-fits-all approach. We don't know who will or won't respond, so we learn next to nothing. Adaptive trials allow us to separate patients on the basis of biomarkers, learn what works, and apply that learning to the next patient. They make it possible to test multiple drugs and combinations, saving a lot of time and money. As new biomarkers are identified, more patients can qualify for inclusion, increasing the number given access to new treatments. Everything is brought together in a continuous learning system, setting a new course that can be replicated for other rare diseases.
It is going to take an international effort to accrue enough patients, to test enough drugs, to explore enough biomarkers to really revolutionize this disease.
It's a pilot, it's a new paradigm, and it will be a model for many other types of cancers if this works. So it's a good learning system for China too.
The innovation comes from a determination by this group to effect great change, and what they call the coalition of the willing, and the desire to serve the needs of patients.
By having the common mission, the egos go out of the window, and what you end up with is pure collaborative intellect.
People always ask me the question, you know, when you do these big things, what makes you think that people will want to participate? Boy, why would you not want to participate when you can change the world?
The end. Thank you.
You can just take questions offline in the interest of time.